Good evening, everyone. I'm Suman Dungal, and we are organizing this free workshop in computational drug designing, where we will be teaching you about the basic steps involved in computational drug designing, where we will be covering the databases as well as a different workflow involved in computational drug designing, structure-based drug designing, ligand-based drug designing, as well as property-based drug designing. We'll be dealing with molecular docking as well, and finally, we will be explaining you about in silico pharmacokinetics, where you can easily screen the drug molecule or any sort of compound molecule for different ADMET parameters, drug likeness, and the broader ADMET parameters as well. So I have prepared one presentation as well. So let me present you the basic information about drug designing. Each and everything will be covered over there, and later on we will be doing hands-on training on drug designing, specifically whatever we will be covering on theoretical part. Okay, so let me share the screen first. So let's get it started. So before I move on to the presentation, let me introduce you to the course content that we'll be covering in the I can say presentation. So first of all, we will be dealing about the introduction of computational drug designing, the approaches involved in CADD, the workflow, as well as the preparation of ligand and receptors before the molecular docking, and what actually is molecular docking. We'll be talking about that molecular docking as well, and optimization of binding site. You can say the prediction of binding site, or you can even say either pocket present in the receptor molecule, and then we'll be running docking simulations, and then the validation of docking will be done later on into the hands-on training. So let's get started. So first of all, computational drug designing. So computational drug designing is a technique where we use different sort of computational tools, online tools, as well as different softwares to discover, or you can say screen or further analyze compound or drug molecules to find their bioactivity. Okay, so it's a very rapid and automatic process because we use different computational tools. It functions based on their algorithms, so it also reduces the cost and save much time as well, because in case of traditional drug designing and discovery, people used to spend a lot amount of time as well as money. So we can easily reduce that cost as well as financial burden using computational tools. Okay, so it also minimizes the chances in failure in different phases of drug development. So we all know that in case of drug you can say development, there are a lot of clinical trials involved into the process, so it takes a lot of time. So if you use computational tools, or if you use computational drug designing approaches, then you can eliminate those steps, as well as in the preliminary step, if you get the information about do or don'ts in drugs or even compound molecule, you can easily eliminate those failures. Okay, so to be a drug molecule, it needs to move on to different steps, either from the clinical trials or the advanced steps. So you can easily screen that molecule by using computational drug design. Later on, you will be moving on to the clinical trials with much confidence as well. So in case of computational drug designing, we actually use two different approaches. One is a structure-based drug designing, and another is ligand-based drug designing. So they are based on whether the structure information of receptor or ligand molecule is present or given or not given. Okay, so basically in case of structure-based drug designing, the structure of target protein, or you can say that the target receptor, is well known, so you can start from there. In case of ligand-based drug designing, the structure of target protein is not known, so you need to start from the ligand molecule. So in between this structure-based drug designing and ligand-based drug designing, there is another approach as well, which is widely known as property-based drug designing. So it is focused on actually the property of ligand, or you can say drug molecules, basically ADMET parameters, in silico pharmacokinetics, and everything. You can use either in silico or in vitro methods for the pharmacokinetic property, ADMET parameters, what we call it. So although there are basically two different approaches, you can also add property based drug designing as well. Okay, so let's move ahead. So it's a basic workflow involved in computational drug designing. First of all, you need to identify the targets. Okay, target means receptor, where you can get the active site, or you can say the pockets. Okay, first of all, you need to identify the receptor, actually the target where your ligand molecule is going to get interacted. Okay, so that is the target. So once the target is identified, that means if the structure of target is known, okay, for example, the target can be a protein or DNA molecule, nucleic acid, what you call it. So once the information is present, either if the 3D structure is present, then you can move on to structure optimization, and then you can predict the binding site. We call it active site as well. So and then you can get the fully functional target structure. Okay, so if the 3D structure is not present in the database, in case of protein, you can say protein database. If the structure is fully not solved, or if it is not available into the databases, then you can go through molecular modeling. Okay, you can say it homology modeling. You can prepare the similar kind of protein, standard protein, and then you can later on optimize their structure. Okay, homology modeling is another important computational approaches in drug designing, where your target, where if you do not have the 3D structure of target, then you can easily do homology modeling. Okay, so once the target structure is validated, or you get the targeted structure, you will then perform molecular docking. Okay, so you need to have a target or protein first, receptor first, and then you need to have a ligand as well. So you can produce ligand from different approaches. For example, de novo ligand designing, what you call it. So ligand designing, you can go through virtual screening as well, or you can go through you can say organic synthesis or any other natural product derivative. You can easily obtain that, or you can even design by yourself, and then you can optimize those optimize the structure of those ligands, and then you can prepare a compound library. Multiple ligands would be there, and you can dock that ligand molecule into the prepared receptor. So the similar pattern will be followed onto the ligand as well. For example, in case of protein, we will be optimizing the structure. So in case of ligand as well, we need to optimize the structure, we need to minimize the structure, we need to minimize the energy of both the protein and ligand, and then we can only dock the molecules. Okay, so docking means we need to prepare the ligand and then drag that ligand into the active site of that protein. Later we'll be presenting in the picture as well. So let's move ahead. So once it is interacted into the once the ligand is interacted into the you can say active site of the protein, there would be forming a lot of complex molecule. You can say a different conformation will be generated actually. So complex means simply the combination of ligand and protein. So once the ligand is interacted into the active site of that protein, it will be generated different sort of conformation. Okay, the molecular docking process would generate different sort of conformation of that particular ligand. So based on the two major factors, we will be ranking them based on, you can say, energy. Okay, minimum binding energy as well as interaction pattern. So the binding energy, as we all know that whenever we perform any kind of work, we use minimum energy to perform that work. Okay, and that would be the efficient work as well. So similar kind of concept would be applied here. So we need to select the minimum binding energy of that ligand molecule into the you can say active site of that protein. So based on the minimum binding energy as well as interaction pattern. So let's talk about interaction pattern here. So interaction pattern means the ligand molecule needs to be interacted with the main or key residues. Okay, key amino acid residues present into the pocket. So once the interaction pattern is validated and the minimum binding energy is present over there, then you can rank that conformation. And Next thing is that there would be involved one statistical factor as well, statistical parameters, what we call as RMST, root mean square deviation. So the root mean square deviation should be less than 2 angstrom. So the confirmation that has minimum binding energy, perfect interaction pattern, and RMST value lesser than 2 angstrom, we can easily select that particular conformation. Okay, so once it is selected, it is called as a potential heat. So before going on to molecular dynamic simulation, we also needs to validate our docking protocol as well. So later on I will be giving you much detailed information about how to validate your docking protocols. Once it is validated, you can move on to molecular dynamic simulation, and then you would be finding the stability of that ligand molecule in the pocket, and that is what we call it molecular dynamic simulation. Actually, it simulates the actual environment present in our body in the in silico method, via in silico method. Okay, so once the stability and everything is determined by using molecular dynamic simulation, we will be going on to different biochemical assays. So in vitro work will be performed here, followed by in vivo as well. So in between that process, we can even do QSR and ADMET analysis. So ADMET analysis, it can be done after biochemical assay or MD simulation, or before that as well. Okay, if you do ADMET analysis before screening this molecule, then you would be getting a perfect result as well. So if the drug likeness and broader ADMET parameters, you can say the parameters are failed for any kind of ligand molecule, you do not need to perform docking as well. Okay, you can easily eliminate those ligand molecules. So once the ADMET parameter is fulfilled, then you can continue the docking, and later on, once it is you can say validated by molecular dynamic simulation, you can again perform ADMET analysis. Okay, for example, in case of being a validated complex, you can go through molecular dynamic simulation. If the molecule is not validated over there, then you can go through derivatization. Okay, you can either perform pharmacophore modeling, what we call it. Okay, in case of pharmacophore modeling, you can either change the, you can say, substituent, or you can say function group present in the compound, okay, to make it more functional, and then you can come back to perform molecular dynamic simulations. Okay, and there is another you can say computational approach as well, what is called it is called as a QSR, that is quantitative structure activity relationship. So based on the structure with the biological assay, okay, we can do QSR analysis as well. You can prepare different models based on their you can say biological activity. Okay, so based on their based on the nature of their bioactivity, you can rank them and then select the best QSR model, and further take that into consideration, or further take that into further analysis as well. And once everything is completed over there, it is called as a lead molecule. Okay, so you can easily take that lead into further analysis, okay, in vivo work or any other clinical trials. Okay, that is the overall view of CADD approach. Let's move forward. So it's a pictorial representation of structure-based drug designing. You can see a ligand molecule and a protein molecule. You can say a receptor molecule. Here it is protein. So the ligand molecule can be produced from organic synthesis or Bartol screening. So organic synthesis, you can go through natural product screening. You can isolate those compounds. You can derivatize. They can prepare the derivative as well. Okay, and then you can get the functional molecule. Okay, that is the ligand molecule. Okay, so in case of protein, you can either get it from the databases, for example, protein database or UniProt or any other protein databases. So the major one are protein database as well as UniProt. So you can get the information, get that molecule from that database, or you can also perform homology modeling if the 3D structure is not available in the database. Okay, so you can prepare the protein by this two method. Once the protein is prepared and the ligand is present over there, you can easily dock that ligand molecule into the active site of this protein. Okay, and then it will form a complex molecule, and that can be further proceeded to form potential drug. So let's talk about molecular docking now. So molecular docking, as the name suggests it, dock means to drag a ligand molecule into the pocket of the receptor or active site of the receptor. Okay, so it's an in silico method actually to and used to find the interaction of ligand molecules with the active site residues, okay, or you can say amino acid residues present in the active site of the receptor. And once it is docked over there, it will form a biologically stable complex. It is determined by the minimum binding energy and the interaction pattern that I have already told you. And the major objective of molecular docking is to find the best conformation. Okay, I have already told you about molecular docking. So the tools, or you can say docking tools, they will give you different conformation of ligand molecule in the active site of the protein. So based on two major things that we have already discussed, we will select the best conformation. So the molecular docking is done to find the best conformation of a ligand molecule in the active site of a protein or receptor, what you can call it. Okay, and it uses different it uses basic tools such as sort algorithm as well as scoring function. So scoring function, you can call it there are different force field applied. You need to apply some external forces to make this molecule docked into the active site. So we call it a scoring function, and it will give some score as well in the form of energy. Okay, and then next is the tools used for molecular docking. They run on different kind of algorithms. Okay, you can say genetic algorithm, Lamarckism genetic algorithm, or whatever else. Okay, they are based on this kind of algorithms, this kind of formulas where they are prepared. Okay, and based on where we perform molecular docking. So it's an example of, it is a picture of molecular docking where you can see receptor protein and ligand, and it is both of them are integrated, or ligand is docked into the receptor to form a stable complex. Let's talk about the technique now. So there are four major techniques involved in molecular docking. You can make ligand rigid as well as receptor rigid in one case, or you can also make them both flexible, or one of them flexible, one of them rigid, and so on. Okay, so in this way you can perform molecular docking in different ways. So it is based on what type of ligand molecule you are using and what type of protein molecule you are actually using as a receptor. Okay, so next is different tools that we use for molecular docking. Okay, so autodock vena or autodock tools, we use it. They are freely available tools. Okay, they are freely available in we can get that online, and they are free for academic users. Okay, and they are supported in different sort of platforms such as Mac, Linux, Windows, whatever you are using, and they are based on the genetic algorithm, Lamarckism genetic algorithm especially, and they use a specific force field. It is known as autodock. Okay, autodock has their own force field. Okay, that's how it is done. And there is another commercial you can say program tool that has been used for many years in a professional setup. It is called Gold. Okay, it is available commercially. You need to purchase with some amount of amount. You need to put on some amount over there, and it is used in different sort of platform. You can get the information over there. Okay, the mostly used you can say tool is autodock vena, autodock, whatever we call it. Okay, there are two, autodock and autodock tools, and autodock vena are different thing. Okay, autodock it has maybe 1.4 or something like that, and autodock vena is the better you can say tool than autodock tools. So next is ADMET parameter. So ADMET, we can call it a in silico pharmacokinetics. Okay, where we analyze the property of compound or a drug molecule. Okay, so basically the drug likeness nature would be analyzed over here, and it falls on the property based drug designing. I've already told you, in case of property based drug designing, we will be examining the property of the compound molecules, where we will be screening their broader ADMET parameters. Okay, to consider them as a drug molecule into the further screening. So Any kind of molecule, any kind of compound molecules or any other molecules is considered as a drug. Okay, considered as a drug once it fulfill different guideline values. Okay, there has been given different guideline values for a compound to be considered as a drug. Actually, ADMET parameters are analyzed for orally administered drug only, because FDA had approved that, or FDA had said that orally administered oral administration is the best route, or oral route are the best route for drug administration, and it is most active, or you can say most reliable, or you can say most followed method as well. So in case of any kind of molecules to be considered as a drug, they have given different you can say guideline values. Among them, Lipinski rule of 5 is one of them. So it has 5 different guideline value. If any kind of compound or drug molecules follow Lipinski rule of 5, then they are considered as a drug. So once the compound has this kind of parameters fulfilled, then you can consider that as a drug molecule, or you can say drug like molecules. Actually not a drug, but drug like molecule. Okay, you can screen both the compounds as well as drug, because there are a lot of mutations, there are a lot of different kind of variations, or you can say a lot of other, we can say MDR or whatever things. Okay, so you need to equally screen the compounds as well as drug. So drug repurposing are quite parallelly done in this world because of different kind of toxicity or any kind of environmental factor. Okay, so you need to screen, we can easily screen the drug as well, okay, for their effectiveness. So in case of Lipinski rule of 5, you can see there are 5 different parameters where compound or any drug molecules needs to be followed to consider as a drug like nature. Okay, so first guideline value is it should have less than 5 hydrogen bond donor. Next is less than 10 hydrogen bond receptor needs to be there. Molecular weight needs to be less than 500 Daltons. The lipophilicity value should not be above 5, and molar refractivity should to be between 40 to 130. Okay, so if a compound molecule has this parameter fulfilled, then you can consider that compound as a drug like in nature. Okay, so it will just give a drug likeness nature. The broader parameters or broader ADMET parameters needs to be studied further. Okay, so ADMET stands for absorption, distribution, metabolism, excretion, and toxicity of the drug. Okay, they are very vital parameters for pharmacokinetic study. So you can see the figure over here at the right hand side. So absorption that take place in the erythrocyte or enterocyte, sorry, or small intestine as well. So distribution, it happens throughout the body. Metabolism is particularly focused on liver. So liver is a great site for metabolism. Cytochrome are present over there. Okay, so next is excretion. Excretion, the major excretory part of you can say drug is kidney, and that is how the ADMET process runs into our body. So now let's talk about in silico pharmacokinetics, where we can use different tools for in silico pharmacokinetics prediction, or you can say ADMET prediction. So you can use Swiss ADME for ADMET prediction. You can use FAB-DROG4 or multiple other tools for ADMET prediction. Okay, I personally recommend using Swiss ADME and PKCSM. So Swiss ADME is used for analyzing the drug likeness nature of any kind of compound molecule, and PKCSM, it calculates the broader, you can say quantitative, the quantitative analysis would be done through PKCSM. It calculates all of the basic you can say parameters of ADMET. Okay, so parameters involved in ADMET, and you can easily analyze that compound or your experimental drug as well. Okay, so that is these are the online tools. Okay, you can easily use it. So Swiss ADME and PKCSM are both online tools. You do not need to download them, they are not the software. Okay, you can easily do it. Next is bioactivity prediction. So if you have a ligand molecule, okay, then if you do not know the bioactivity of that ligand, then you can easily use this tool, PASS online tool, to predict the bioactivity. Okay, so it is based on the structure information of ligand molecule. Okay, you can say NMR has been done, or NMR is done for the spectral information is gathered through NMR data. Okay, and based on that data, the bioactivity prediction is done using pass. Okay, it gives a result in the basis of probability. Okay, 0 to 1 range, and the average accuracy of this tool is 95%. So you can trust that. Okay, it's reliable in nature as well. So here I have given one example, aspirin. Okay, aspirin is a immunoinflammatory or in inflammatory, okay, anti-inflammatory drug as well, actually, okay, antipyretic in nature as well. So aspirin, the bioactivity has been predicted by pass online. You can get the you can see the result of aspirin bioactivity prediction using pass. So you can see the P and PI value. Okay, PA stands for probability of being active and PI stands for probability of being inactive. So here you can see 0.9, the ranges are above 0.9, so the guideline value is 0.5. So you need to you need to select those you can say bioactivity of a particular ligand whose PA value is over 0.5. Okay, you can easily select those you can say bioactivity or similar kind of receptors. For example, fibrinolytic the fibrinolytic activity has been shown by aspirin. That means you can easily dock aspirin into fibrinolytic receptor. Okay, similarly antipyretic and multiple other you can say bioactivity has been given over there. Okay, so next is let's talk about databases, okay, involved in CADD approach. So in case of databases, we need to talk about the protein database as well, or can say receptor database as well as ligand database. So in case of receptor, we actually use a protein or enzyme basically. So for protein and enzyme, you can go through protein data bank. Okay, you can get the information of protein or any enzyme. You can easily download them and go for the molecular docking thing. Okay, and for ligand, you can use PopChem and DrugBank. So in case of protein, you can also use UniProt as well. For ligand, you can use PopChem, DrugBank, Zinc, whatever. Okay, the link has been given incorrect, so skip it. Next is for structure and file format conversion of ligands. You can either use ChemInfo, OpenBabel, ChemDraw. Okay, so you can easily sketch using ChemDraw. You can download OpenBabel. OpenBabel and ChemDraw are software. Okay, in case of OpenBabel, you can easily convert the file formats of ligand or even protein, and you can even use OpenBabel for energy minimization of ligand and protein as well, especially the ligand. And in case of ChemDraw, you can easily sketch the structure of any kind of molecules, any kind of compound molecules. In DrugBank, it falls under it is a database for ligand. Okay, so let's move ahead. So let's talk about the prediction of binding site, or you can say the active site prediction. So we can use two different tools here. I've given two tools. One is CASP and another is PDB-SOM. Okay, so in case of PDB-SOM, you can easily analyze the active site residues, or you can say the pocket of a particular receptor. Okay, a particular receptor, and the LigProt analysis. Actually, PDB-SOM it incorporates the LigProt data. So LigProt is an external software. You can easily download it and then see the you can say this kind of images would be generated. You can see the active site residues as well as pocket of any kind of protein molecule, okay, or any enzyme molecule. So you can easily use PDB-SOM for the standard protein only. Okay, not for the non-standard. So for non-standard, I'll let you know about how we can analyze the active site of non-standard protein. So next is CASP. CASP also predict the pocket. Okay, you can see the image over there. So if the 3D structure of any kind of protein is known, and if it has a parent ligand or tails over there, then you can easily use PDB-SOM. But if there is no any parent ligand present, or if the protein has not been fully you can say solved, then in case of that, you need to go through homology modeling. you need to prepare your own model. You need to validate that model as well, superimposition, as well as you can either perform superimposition, or you can draw Ramachandran plot, the mole probability score, and a lot of other scores needs to be validated over there. And once it is completed, you can go through molecular dynamic simulation to validate the stability of that model. And then you can go through different online tools to predict the pocket, okay, in this case. But another way to identify the active site is to do multiple sequence alignment of that particular protein. So for example, if you have a protein obtained from a human, then you can similarly get the protein sequence you can get the protein sequence from NCBI or any other databases. Okay, for example, let me make it clear first. So if you have a protein that is a protein, okay, if it has been extracted from human, you need to go through NCBI or any other two and get the protein sequence first. Okay, and similarly you need to get two or three different sequences, closely related sequence of human. Okay, for example, if a protein is obtained from human, then the same protein needs to be obtained from the close related organism of human, for example mouse or monkey. Okay, so once the same protein structure for different organism is present, you can just do molecular dynamic or molecular you can just do multiple sequence alignment. And then once the result is there, okay, you can just check the conserved region. Okay, so you can check the conserved region, and you just need to analyze the longest conserved region present on there. Okay, once you identify the longest you can say longest conserved region, that longest conserved region is considered as an active site. So in this way you can easily predict, okay, the active site for non-standard protein. So next is file format. So in case of ligand and protein, we use different sort of file formats. In case of ligand, we use a smile strings, STF file, mol or mol to even. Okay, so these are for ligand file formats. And in case of protein, we use pdb file. .pdb would be the extension. Okay, and next is pdbqt. So pdbqt stands for pdb with partial charge is q and autodoc 4 atom types t. Okay, so pdbqt file is generated right after molecular docking. Okay, next is molecular visualization tool. So if when you are looking for the entire structure of receptor protein, or you can say enzyme, you can use different sort of tools, computational tools, you can say online tools or software, you can call it. UCSF Chimera is one of them. It has the best graphic quality as well. So you can use UCSF Chimera. You can either use PyMol or Discovery Studio Visualizer as well. Apart from that, you can even use CN3D or any other visualization tools. Okay, so in this particular workshop we will be dealing with UCSF Chimera. So UCSF Chimera is also a simple tool, okay, where you can incorporate your Autodoc VNA and you can easily perform molecular docking. So that is the basic thing involved in this workshop. Okay, so this is me. So we are focused on different sort of programs as well as product development in Nepomics. Let's see it. So in Nepomics, we organize different sort of workshop and webinars, and we have already organized it. You can get the information in the YouTube as well. You can get the webinars over there, and then if you follow our social media, time to time we will be organizing different sort of workshop, as well as intensive program would be is already been used or already been organized. It's already onto the ninth batch this year. Okay, so we are doing these things basically focused on computational drug designing as well as basic to competent bioinformatic analysis. Okay, any sort of bioinformatic analysis, sequence based analysis. And next is we are also focused on research and development of natural product derivatives. We have already developed different personal care product. The major product are natural mosquito repellent, natural room fresheners, anti-hair loss oil, and many other, actually 5 different products are already listed into the online platforms, and we are doing it commercially. So that is all about this workshop. Now in the second part of this workshop, we will be dealing with hands-on training on computational tools. Okay, using different computational tools, we will be screening compound molecule, different compound molecule, and we will be performing molecular docking as well as in silico pharmacokinetics using Swiss ADME and PKCSM. Okay, so that's it for the presentation. So we will be moving on to hands-on training. Thank you. So before moving on to the different databases, I have mentioned the link of the databases, particularly PubChem, you can see Uniprot, PDV, and other as well. So you can just use this link to get the databases as well as some tools. So you can see a Chimera here. Okay, so you can download using this link based on your system, whether you are using Windows or Mac. You can easily download Chimera over there, and then later on we will be doing molecular visualization as well as molecular docking through Chimera. Okay, so next is Autodoc Vina. So I have given the link to download Autodoc Vina as well. You just need to download vina.exe. No need to download scripts for this workshop. Okay, you just need to download vina.exe, and you can give the pathway later on into the Chimera, and you can easily perform molecular docking. And later on, this is a link for Swiss ADME. We will be using this link to get the Swiss ADME platform to screen the drug likeness nature of compound molecule or any other drug molecule. This is the link for PKCSM. We will be analyzing the broader ADMET parameter of any kind of drug or compound molecule using PKCSM, and this is for protein data bank, and this is for Uniprot. Both of them are protein databases, and okay, this is for PubChem. So there are different databases used for ligand. One of them is PubChem. Next is DrugBank or Zinc. So I've just used PubChem over here. You can easily get that information. Okay, so now let's go to select our ligand as well as protein molecule. So today we are going to use aspirin as a ligand and cyclooxygenase receptor, COX2 receptor particularly, as a important receptor for any kind of immunoinflammatory or antipyretic drug molecule, particularly aspirin. Okay, so let's do it. So first of all, we will be going through protein database and then download COX receptor cyclooxygenase, and then we will be searching for aspirin molecule, which is antipyretic in nature as well as anti-inflammatory in nature. So it is perfectly fitted into the COX receptor. Actually, COX is a perfect receptor for this kind of molecule. Okay, taking into consideration of that fact, we will be docking aspirin in the active site of COX molecule. So let's proceed further to see the protein molecule. Okay, to download it and then get the maximum you can say output result of COX there in the PDB. Let's see it. And there are different parameters used to select the protein molecule. Okay, because if you type cyclooxygenase over there, you'll be getting a lot of information in the PDB. You can easily filter out whatever you want. For example, if I'm working on human cyclooxygenase, I can easily filter it out. The filters are present on the left hand side. Okay, you can do that. And then there are other guideline value that needs to be fulfilled as well. Okay, to select a particular protein molecule. So here you can see R free value, resolution, class score, Ramachandran outlier, side chain outlier, RG outlier, ligand fit scores, and these are the guideline value to consider any kind of best protein molecule present in the database. Okay, that means you'll be getting different sort of structure for similar protein. So this guideline value can easily help you to screen. For example, the R free value needs to be 0.202, 0.25. okay, later on we'll be screening it. Okay, you can just copy this, you can just get the information. Okay, let me go to PDB. Okay, protein data bank now. So this is a homepage of PDB. So I've already told you, now we are going to sort cyclooxygenase. Okay, so I've already done the work on cyclooxygenase. So I know the PDB ID of cyclooxygenase, that is 6COX. I'm particularly focused on 6COX in this particular you can say workshop. But if you don't know the PDB ID, then you can easily type cyclooxygenase over there and you can get the information. Okay, so for now I'll be just typing 6COX. Okay, 6COX. Okay, let's see, it should be in a capital letter. Actually it has it's a PDB ID as well. So it has PDB ID, the 3-letter system is there and one number. Okay, so this is the PDB code. Actually it's a cyclooxygenase 2 receptor. It is called prostaglandin synthesis 2. So I was talking about a filter, so I'll let you know once it is screened. Okay, so let's see the information of cyclooxygenase here. So the PDB ID is 6COX and you can get the information. Here is a inhibitor called SC558 present in this complex. Okay, so next is the classification of this particular protein is oxidoreductase, and it has been obtained from Mus muscarinus, and it has been expressed in this organism. And next you can see there is no any mutation present over there, and this protein has been solved through X-ray diffraction. So you need to select the protein molecule that has been solved through X-ray or NMR. Okay, you can eliminate or you can ignore X-ray or any other in silico method. Okay, and the resolution. So now here it comes the guideline value. You can just go through the guideline value as well. For example, the previously I was telling you the resolution needs to be less than 2 angstrom. So here it is 2.80. Ignore this right now because we are just practicing it. So but in your actual research you need to select the protein molecules where the resolution is lesser than 2 angstrom. Okay, so there is another parameter as well, what we call it R value. Okay, R value or R factor as well. The R value or R factor, R free value needs to be 0.20 to 0.25. Okay, R free value. Okay, R free, where is R free value? R free value. Okay, it is greater than this. So actually the value is not satisfying right now, but you need to select the best one. Okay, and next is the class score needs to be less than 10. So let's see the class score. It's 35. It is greater here. Okay, if the class score is greater, then you'd be expecting some classes or some kind of classes present into the residues in that particular protein. So to eliminate that kind of classes, you need to select the protein molecule that has a class score of less than 10. And next is Ramachandran outlier. So we all know the Ramachandran plot is actually used to validate the protein structure. Okay, so in case of here the Ramachandran outlier needs to be 1%. In this particular protein it's 5.8%. So actually it is not favorable. Okay, so you need to select such kind of protein which has RMST Ramachandran outlier less than 1%. Next is side chain outlier needs to be less than 5. It is 13% here, so it is also bad here. And RSRG, where is RSRG? So they haven't given. Okay, the RSRG value. So whenever you see RSRG value over there, it needs to be less than 5. So next is RSRG. So let's talk about the RSRG value. So RSRG value implies the real space R value J score. So it's a statistically significant G value as well. It measures the fit of model to the local electron density. Okay, it actually measures the fitness of the model. So you can easily screen that RSRG value as well. So the value needs to be less than 5%. Okay, next is ligand fit score. So we cannot see ligand fit score as well, so it needs to be closer to 1. So next is, you can see the literature as well, so you can read the data, you can read the abstract, you can read the paper to get the information about cyclooxygenase. Okay, this particular cyclooxygenase. And on the left hand side you can see the structure of this protein, and just below that you can see the global symmetry. It's a cyclic protein and it is a homodimer. Okay, that means it has two chain, and both the chain are similar in nature. Okay, so while performing molecular docking you can just select one of them. And next is, when you go through it you can see the chain and everything. Okay, and this is the most important part that is important for molecular docking. Okay, so small molecules, the parent ligands, what we call it. Okay, there are three different parent ligands present in this particular protein, particular complex, you can say it: hem, S58 and NAG. Actually S58 is considered as a inhibitor. They have already performed this you can say research work, and hem and NAG are cofactor. Okay, so actually it has three different active site region, or three different pockets present in this particular protein. So you can either dock your we are docking aspirin, so let's say aspirin. So you can either dock aspirin hem region, S58 region, or NAG region. Okay, the most active part is S58 because S58 directly acts as an inhibitor. So you can eliminate or you can remove S58 and dock your aspirin. Okay, later we will be doing that. And this is the information that we can get from we can say PDB. So now let's talk about let's talk about addition of filters. Okay, let me let me copy this, and then let me tell you how filters can be applied. Okay. Okay, here you can see at the left hand side you can see the refinement, so you can easily select. Okay, if you are searching for cyclooxygenase receptor of human, then you can mark it. You can also go for experimental method, wherever whatever you want to. For example, I have already told you X-ray diffraction and NMR are better options, so you can go through electron crystallography is another widely used approach. You can also select that, but do not select electron microscopy. Okay, electron measure the 3D structure can be solved through X-ray diffraction and NMR. So these are the most important methods. You can select one of them and go through the analysis. Okay, next is you can also put filters on you can say resolution. So resolution needs to be 2 angstrom, less than 2 angstrom. You can do that. You can also go through the latest data wherever you want, and you can go through the classification. You can go through the membrane protein annotation datas. You can go through symmetry. You can go through other different information. So based on that you can select your best protein. Okay, this is all about PDB. Now we will be downloading our structure. Okay, cyclooxygenase. So in case of COX, you can just go through it, and here you can see the download files. You can easily download the legacy pdb format. Okay, you can just click here and you can just download pdb, and please make one you can say a folder on your desktop or any other wherever you want to do. Okay, for example, in my case I have prepared one folder in the name of workshop, so I'll be using each and every downloaded or extracted file in that particular folder, and later on it will be used for molecular docking, and it would be much easier to you can say get those files into that particular folder. Okay, it would make your work simple. It would make your work more efficient as well, and more you can say easily the file can be easily available in the particular folder. So here you can easily download it. Okay, so that's how we can select our protein. Okay, once we select our protein, then we will go through further analysis. So it is it. Now let's talk about the ligand. So for ligand, for any kind of ligand databases. You can use PopChem, or you can even use DropBank or Zinc databases. Now I have opened the home page of PopChem. So as we have already downloaded the cyclooxygenase receptor, now we have to focus on ligand. So for cyclooxygenase ligand, in case of cyclooxygenase, aspirin would be a best ligand option. So let's go to see the properties of aspirin and copy, or you can say you can even download the STF file here as well. Let's see the information first. It's aspirin. Let's open it. So you can see the structure of aspirin, you can see the molecular formula, you can see the synonyms, you can see the molecular weight and lot of other information for aspirin molecule. Okay, it's a two-dimensional structure. You can get a three-dimensional as well. Okay, this is a three-dimensional structure of aspirin, and later on you can see the identifier. So it's a file format. Actually I've already presented there. So a smiley string, so it's very important during analysis. You can easily copy this smiley string, and later we will be working on to onto the you can say energy minimization and later a step of molecular docking for this particular aspirin. Okay, I've copied this small string of aspirin to use it further. You can see the molecular formula, you can see the CAS ID as well. Okay, it is used in commercial aspect. You can see EC number. It is particularly used for UNI number and EC numbers. Their UN number, they are particularly used to prepare MSDS, material safety data sets, and it is widely used for toxicity analysis as well. And there are different information you can get it. For example, the spectral information you can get it from here. Okay, the spectra of aspirin, NMR spectras. You can get a lot of other information. You can say pharmacology of aspirin. You can say cyclooxygenase. It has been mentioned over there. So we will be using cyclooxygenase into the analysis, into the molecular docking analysis. Okay, next is the uses and the pharmacokinetics, pharmacodynamics, and everything is present over there. You can see the toxicity, you can see the safety and hazards as well. You can see the literature from where they have worked, or the multiple researches that has been conducted in aspirin, the patents they have got for aspirin, different sort of aspirins, or even the derivative of aspirin as well. You can see interaction and pathways, you can see biological test results, and a lot of other information. You can get it from PopChem or any other databases. Here you can see the PopChem. Okay, apart from PopChem, you can also use DropBank or Zinc database for any kind of compound. PopChem is the best one, but if you are screening any drug molecule, then you can go through DropBank, and then you can easily get the information. Okay, that's it. That is the information that we can get from PopChem. So I've already copied okay, the identifier. Okay, now we'll be moving on to further analysis. So preparing the structure of aspirin, and then energy minimization and everything. Okay, so before that, let's do the bioactivity prediction first, and then we will be moving on to further analysis. Okay, so let's see. I've already copied the smiley string. Okay, let me copy it again. So because in case of activity prediction, it is required actually. So let's do the bioactivity prediction of aspirin, whether it gives cyclooxygenase or any other you can say receptor or biological property. So let's confirm it from pass online tool. I've already told you about pass online tool, so you can easily get the bioactivity of any kind of compound molecules to pass online, and it extract spectral information of any drug molecule or compound molecule to give the bioactivity prediction. So let's move on to pass online now. So this is actually the interface of pass. So I've already logged in with my account, so here my name has been written over there. So if you haven't registered yet, then you just need to register by giving your information over there, your name, your affiliation and everything. And then once it is validated, they will send you the username and password into your email, and you can come back and then log in and you can get this information. Okay, now let's predict the bioactivity of aspirin. So once you click on there, you can see the smile string option. You can just paste the smile string. I've already copied this. Let's see the prediction now. So now here it is. Because of some kind of internet issue, maybe it was you can see lingering. So let's see the result. Okay, so I've already pasted this result into the presentation as well. So you can see the PA value is over 0.9. So you can easily select any of those receptors, or any of this biological property can be further analyzed to select the, you can say, receptor. For example, fibrinolytic receptor can be selected, antipyretic. Okay, you can see all the different kind of things over there. Okay, so you can easily select them, okay, based on the PA value. The PA value needs to be 0.5, over 0.5. Okay, so that is how you can predict the bioactivity of any kind of molecule. That's about pass online. Now let's go for active site prediction. Okay, so the active site prediction is done through PDB-SOM as well as CASP. Currently CASP is not working, so we'll be analyzing the active site residues through PDB-SOM. So we have already you can say taken protein. Okay, protein cyclooxygenase as a protein. So let's see what kind of active site residues are present in a cyclooxygenase. Okay, 6CX particularly. Okay, let's see. Now let's talk about the active site prediction using PDB-SOM. Okay, it is a working interface of PDB-SOM. So previously it used to give the direct result. Nowadays it has been converted to some kind of this you can see the workflow. So once you click onto PDB-SOM generate, you will be moving on to this page where you need to upload your you can say PDB file formats, and then you need to give your email address as well. Once you are done with that, you will be getting information of your particular protein. Okay, the active site prediction would be predicted by using your own software, by using the own interface, and then they will be they will be selecting the result in your email. Okay, so later on I will talk about that as well. So I will talk about how the email how the you can say active site prediction result will be given to the email, or how you can actually analyze that. Okay, let's talk about it. So okay, So this is the interface of PDB-SOM. Actually, it used to be the interface below. You can easily copy this link, and then you can get this interface as well. Maybe I'm not sure about it. You can use this, or you can go through the previous steps, and then they will send the results through email as well. Okay, so let me type the PDB ID of my protein of interest, that is 6COX. So let's see how they can predict the active site residues. Okay, let's see. So 6COX is oxidoreductase. You can read the information present over there. Let's focus on the active site only now. Here you can see the protein chains A and B. We all know that it is a homodimer. So next is there are we have already seen on PDB as well. There were three different ligand molecules, especially S58, specifically S58, HEM, and NAZ. So that there are two S58. That means in the two chain there are particularly two. There are two chains. So one chain consists of S58, one of S58, and another is on next chain. Okay, so that in total gives about 2 S5, and in total there are 2 HEM as well as NAG. So in one chain there are 3 NAG molecules. So we are just let's see S58 region only right now. You can go through HEM and NAG later as well. Let's see S58. So you can see the lead product has been integrated over there. You can see S58 region, or you can say S58 is present over here. This is the molecule, this is a ligand molecule, and these all are the you can say interacted amino acid residues. So these are specifically those amino acids where hydrogen bond is formed, and other sites and residues are present over there. So let's you can just zoom it and get the information. Okay, to see the interaction on a broader way, you can just click on to list of interaction. Okay, if you go through, sorry, okay, list of interaction. Okay, now you can see the hydrogen bond and all the different interaction as well. So in case of molecular docking, we need to consider the hydrophilic interaction, that is hydrogen bond specifically, because they are biologically significant in nature, and there are other hydrophobic interaction as well that should not be ignored, but actually we look for the hydrophilic interaction. Okay, so there are 1 2 3 4 5 hydrogen bond. Okay, the ligand has been interacted with this pocket with the amino acid residues, these amino acid residues forming hydrogen bond. So histidine 90, it has formed hydrogen bond with histidine 19, arginine 120, glutamine 192, leucine 352, and serine 353, and there are other different hydrophobic interaction. Okay, all there are 75 hydrophobic interaction and 5 different hydrogen bond interaction. Okay, later on we will be confirming this information through molecular docking as well. Okay, so actually we will be performing molecular docking through aspirin, but you can redock this molecule. Okay, and then once you get this information, you can see, okay, I've done that, and that is what needs to be done. Okay, so let's see. Now let's move on to chimera and process the molecular docking thing. Okay, preparing, first of all we will be preparing ligand molecule, we will be preparing protein molecule, and we will be performing molecular docking simulations. Okay, let's see. So now let's do ADMET and methods of aspirin molecule. So in that case you will just need to copy the small string of aspirin. So here I have already copied it. Let's proceed further. So it's a small string of aspirin. You just need to run it. Okay, so once you press the run, you'll get a result on the bottom hand side. So here is the result. So you can see the molecular weight, the structure as well, and different other parameters, different other values of those parameters has been mentioned over there. So we just look for the drug likeness nature, especially the Lipinski rule of 5. So let's start with molecular weight. So molecular weight in the guideline, you can see we have already discussed about those as well. So the molecular weight needs to be less than 500 Dalton. It has been satisfied. hydrogen bond donor, hydrogen bond acceptance to be less than 5 and 10, so it has also been satisfied here. Molar refractivity, the value lies in between, lies in between 40 and 130, so it has also been satisfied here. And log p value, you can go through consensus log p value, that is 1.28. so these all are satisfied for aspirin. So you can also get the summary of Lipinski rule of 5 model here. There is no any violation present. So there just like that of Lipinski, there are different other models as well, but Lipinski rule of 5 is a worldwide accepted model, and it is also considered as a gold standard in drug likeness analysis. And apart from that, you can also see the qualitative analysis of pharmacokinetics properties, GI absorption, blood-brain barrier permeability. Either it is substrate or inhibitor of glycoprotein, cytochrome inhibitors, or any other things. Okay, these all are the pharmacokinetic property, you know? Okay, and apart from that, you can also see if it is lead likeness. Okay, there is if the aspirin is further taken into further taken into consideration of lead molecule or not. So because of its molecular weight, it has not been considered as a lead molecule. However, it is already FDA approved drug, so it is not an hard and fast rule in case of any kind of molecule in the screening. But you just need to you just need to focus on the broader parameters. Okay, but if any of the guideline value has not been fulfilled, then you just don't need to hang out there. Okay, you can just take into further consideration. It can be modified later, or even in the original state as well. The broader parameters can be you can say better in the response in vivo or in vitro work. So you just need to consider all of these things. Okay, it's not hard and fast rule in case of here. So we just so here you can see the Swiss ADME model, or Swiss ADME just predict the qualitative analysis of this compound molecule or drug molecule. So for broader activities or broader analysis, you can use PKCSM. So later on we will do PKCSM as well. Okay, so in case of PKCSM, you will be getting the results in the form of numeral numbers. Okay, in the form of numbers. That means a quantitative analysis will be done through PKCSM. So you can compare the result with the theoretical model as well as a number of different drugs. Okay, that has been approved by FDA. So they have given their pharmacokinetics properties or guideline values, or you can say the experimental data has been given for different drugs and compounds, so you can easily compare that with your particular compound. Okay, so in this way we can analyze the data. Now let's move ahead for PKCSM analysis. Okay, so it is a workflow. You can say it is a workstation, or you can say home page of PKCSM. So you can just go through the theoretical part. Okay, if you click onto the theory, you will get one PDF file, so you can easily compare the result for those different models that they have produced here. Okay, you can compare the result. Just click onto PKCSM, so you can just copy and paste your smart string of your ligand molecule, and you can predict this ADMET parameters individually or as a whole as well. So let me do as a whole. Okay, so not just ADMET, not just absorption, distribution, metabolism, excretion, and toxicity individually, but let's analyze as a whole. So if you click onto ADMET, it will give the result, and you can easily compare those results. Okay, and in the standard, you can say guideline value given by PKCSM. Okay, they have analyzed millions of compound, and they have produced this data. Okay, so it's a reliable as well. So in this workstation, you can get the quantitative data of different ADMET parameters. For example, the absorption, it consists of water solubility, K2 permeability, intestinal absorption, skin permeability, glycoprotein substrate inhibitor property, volume of distribution as a distribution parameter, fraction unbound distribution parameter, blood-brain barrier as a distribution parameter, and different cytochromes are present over there, which are considered as a you can say indicator of in case of metabolism, because cytochrome are present in liver and they function as a metabolism key role in metabolism of any kind of drug molecules. Um, in case of excretion, total clearance and renal OCT2 substrate. If it is if the molecule is substrate of renal OCT2 or not, they will calculate it. And in case of toxicity, MS toxicity, or different parameters would be analyzed. Okay, and you'll get the result in the numeral data will be generated over here. You just need to compare the results. For example, you can go through the theory. Okay, then you can get this different data. You can easily analyze those. For example, in case of CACO2 permeability, the guideline value is given over there. Similarly, you can interpret the result by yourself. Okay, there are multiple one. And apart from PKCSM, they also have developed one AI tool. You can say deep PK, what they call it. Okay, you can also use this deep PK and analyze the data. Okay, so that's it for PKC. That's it for pharmacokinetic analysis, and it's very important during drug screening. Okay, so that's it. So now let's do molecular docking of aspirin and 6-COX cyclooxygenase receptor. We have already downloaded 6-COX from PDB, and we have also copied the smiley string of aspirin in PubChem. So let's build the ligand first. Okay, so for that you need to go to tools, structure editing, and then build a structure. Okay, so here you need to select a smiley string. So I've already copied the smiley string of aspirin, so I'm going to paste it. Okay, so this is a smiley string of aspirin. You can rename the residue name as well. For example, here it is UNK. You can name according to the name of your ligand. So I'm leaving it default. Okay, no need to change. So make sure that you write a different you can say 3-letter code than that of we can say amino acid residue. So if it resembles, then the system will not detect your molecule. Okay, so better write a, instead of writing ASP, you can write APS or something like that in case of aspirin. Okay, so once you do this, you just need to press apply, and then you will get the molecule in the background. So let's wait for the result. Okay, please wait, because of some kind of interruption it is not showing. So let me check it again. Okay, smiley string is perfect. Okay, okay, let me apply it again. Okay, now you can see the aspirin on the background. Okay, so here it is the structure of 3D structure of aspirin. So now we need to minimize its you can say structure. The energy minimization needs to be performed. So for that you need to go to tools. You can go through you need to go to structure editing, and then you can move on to minimize the structure. Okay, so in this case you can just increase the number of steps to minimize it. So larger the number, the time will take the much time greater time will be taken. So you just need to take care of this step as well. So for this workshop we are going to use 100 steps. So in your real research you can increase that to 1000 to 10,000 as well. Okay, so leave it default. Everything would be default in this case. You can change according to your you can say desire as well. So that's it, just press on to minimize. So next the hydrogen will be added to this molecule. So for that you just need to mark this, and then so here it appears protonation state. So as it is not a protein molecule, so we do not have any kind of amino acid residues, so better not to do anything. Just leave it default. If in case of protein, you need to consider whatever you are doing in that particular protein. Okay, so for example in case of protein we will be talking about later actually. So let it be here now. Just press OK. So now the charges has been added. No, not the charges is going to be added in this molecule actually. So in case of small molecules, you can just press castigar and the force field is applied over there. The amber force field is there, so you just need to press it. You need to click on to castigar charges and leave it default and then press OK. So next is the net charge of this molecule has been calculated by Chimera. Okay, you can just press OK, and then you can see the structure would be a little bit twisted at the end to minimize the structure. Okay, look, it is the minimized structure of this particular aspirin molecule. So you need to save this molecule in mol2 format. So I've already allocated one folder here, so I'm going to save it as a prepared aspirin mol2 file. So you can do it, and then you just need to save it. Okay, so finally it has been saved onto the particular folder. Now let's prepare protein in the similar fashion. So let's close the session first. Let's go to file, let's open protein, that is 6COX. okay, here it is. So it's a 6COX molecule. So we have already have got the information of 6COX in the PDB, protein data bank. So it says it has two chain and both the chain are similar in nature. That means it is a homodimer. So you can use one of the chain. So I'm going to use A chain. So let's delete the B chain. Domain bonds and delete. So here is the molecule. Okay, so I've told you about the parent ligand present in this COX receptor. So there are three parent ligand NAG, HEM, and S58. So actually in the experimental data we have already found that S58 acts as an inhibitor in case of cyclooxygenase in this particular cyclooxygenase. So let's focus on that inhibitor, and you can say let's screen aspirin in place of that S58. So let me delete other residues. For example, heme. Heme has been deleted. Now let's go to NAG as well. There are three NAG in this chain, so let's delete it as well. So now you can see just S58 only. Okay, so now we are going to use S58 as a parent ligand, and instead of S58 we are going to dock aspirin molecule. Okay, so for that let's prepare this protein first, energy minimization and everything. So for that you need to go through surface binding analysis and then go to dock prep. Okay, now you can see 6UX has been selected over there. No any non-standard residues, or you can say foreign residues, such as solvents and other things needs to be deleted. So here it has been selected as well. So let it be placed over here, and you can see incomplete side chains. So side chains needs to be deleted in some aspect. If you know the detail about the side chain in particular protein, then you can go through this. If you do not know, then just leave it default. Okay, so next is addition of hydrogen, addition of charges, and you can save that into mol2. I'm not going to save this into mol2, so I'm on marking it. Later on I will save it into PDB format. Okay, in case of ligand you can save into mol2 and then protein into PDB, or you can make it save in both the proteins and ligand can be saved in similar format, for example mol2 or PDB. Okay, better you can save on PDB for protein and mol2 for ligand molecule. Okay, so that's how we can do it. So press OK. Then after, the hydrogen would be added in this step. Okay, so just press it default. You can make it default. Hydrogen would be added into the molecule. So here you can see the protonation states that I've already told you in the ligand as well. So in case of protein, it is very important. If you know, if you are working on the particular residue in the protein, then you can go through the protonation state as well. For example, here it is mentioning histidine as a protonation state residues, but you can change it. Okay, so if you know it, or else leave it default, and then leave it default, just go to press OK. So now you can see we are going to add a charge as well. Here you can see the amber force field is applied and gastric charge are marked. Okay, so let it be here. And now S5, the net charge of S5 has been calculated over here. So press it OK. Now it has been well prepared. You can see the result. It's running there, and once it is completed, it will be shown. Okay, now it's the protein has been well prepared. Now you can save it onto PDB, and then you can save it onto you can save as a prepared protein. You can later use this. Okay, let me save prepared protein. Okay, let me save it. So in the same tab, now let's open ligand molecule as well. Okay, prepared aspirin. Okay, here you can see aspirin has been integrated into this workplace. So let's select. Okay, let's select S5 so that we can prepare a grid box over there. Okay, it's S5, and let's prepare a grid box in this particular region. Okay, so for that we need to go through. Okay, let's go to tools and then surface binding analysis and then Autodoc Beta. So you can see it right now. Right there, so click on to browse. You need to give a output file location as well. The pdbqt file will be generated, so you can name it like aspirin docked in cox. Okay, so let it okay, now it has been given there. Okay, now we have already uploaded our aspirin as well. We need to change the ligand notation as a small string over there. And now let's see, let's resize, let's create a grid box. Let's say make it okay 23. You can write a random number. Okay, I'm writing okay 24 44. Okay, and then let's see 22 and then 24. Okay, now you can see a grid box has been generated. I have used this I have used the dimension of the box as I've already performed it before, so I've used it in the standard way, but you can easily change it. Okay, you can remove the you can drag this box using the you can say using the wheel of your mouse. Okay, in this way you can just you just need to tap on the wheel and then you can drag it. The box will be moved. Okay, and then to increase the size and everything, you can just go through the wheel and then just click onto the wheel and then drag it. Okay, the size will be increase or decrease. Next, you can also increase or decrease the size. You can also increase or decrease the size. You can also increase or decrease the size. For example, let's make it bigger and then allocate it further. Okay, okay, it's a grid box. Okay, I've changed the dimension of grid box. So if I need to move this grid box to the active site region, so I can do this. Okay, so you just need to prepare a grid box in such a way that this molecules is inside the box, and make sure the box is smaller in size. Okay, just to cover the active site residues present in the bucket. Okay, make it as smaller as possible. So here it has been created perfectly. So you can see the aspirin molecule is inside the grid box, and then all of the active site residues are also present. So that is how we can do it. Okay, so that's it. Okay, let's check the dimension and everything. So dimension is here, it is 21, 24 and 49. Okay, and then next is 22, 22 and 20, 28, 22 and 26. So let me change into let's say 23. 23. Okay, make it 24. Okay, make it 44. Okay, 22. Okay, make it 22 as well. Okay, 22, and then 24. Let's say 24. Okay, so you can see it has been perfectly placed inside the grid box, and some of the residues, most of the residues are also been inside it. Okay, so here it is. Now you can easily perform molecular docking. You can run the autodock vina as well now to perform the molecular docking to get the binding energy and everything. Okay, so here you can okay, let me see. Okay, now you need to copy this dimension. Okay, so the dimension of center and size as well, size of box as well, so later you can use it. Okay, you can modify the numbers based on the result obtained after molecular docking. Okay, so here it is. Now next is receptor option. You just need to make it true or false based on this default values. You can also change that into true as well in this step. Ignore non-standard residues, so you can make it true as well, because we are not dealing with any kind of non-standard residues. All of the options are correct. Add hydrogen in chimera, so it's true. If hydrogen has not been added before, then in this step as well you can easily add more charges and remove non-polar hydrogen, more charges and remove lone pairs, ignore water. So so we do not do molecular docking inside the water molecule, so we can just we just need to ignore this water. Ignore chain of non-standard residues. I've already told you about the residues as well. So next is ligand option. So you can see more charges and remove non-polar hydrogen, true. More charges and remove the lone pair. Okay, in this aspect you can go through the options. Okay, so next is advanced option. You can see number of binding modes. So after performing molecular docking, you will be getting 9 different docking poses given by autodock vina here in this case. And next is exhaustiveness of the source. Exhaustiveness means the reliability of this result, so you can mark it to 8. You can change the data, and maximum is the 8, so let's make it to 8 as well. And next is minimum or maximum energy differences, so you can just make it to 3, or you can just increase whatever you want. Okay, so and the default mode, it is selected C, so I'm not going to change it. Okay, next is, so we need to give the pathway of the vina exe. That is when already downloaded. I've already given you the link as well, so you can just go through the browse and then go through the folder where your vina exe file is located. Okay, once you do this, you just need to okay, in this I have forgot to remove this parent ligand, so let me go through residue. So delete this, it hasn't already selected, so let's delete it. So here is it. Now you can run the autodock vina. Okay, so or okay or apply. Okay, do not press okay and apply for a couple of times, or just press one of them, okay or apply. Okay, okay, I pressed okay, so let the vina be running. Okay, now you can see the result has already been obtained. So you can see the binding energy of different conformation. There are 9 different conformations that has been generated, and the binding energy has been resulted to be minus 6.9 kilocalorie per mole, and so on. Okay, and here you can see the RMST lower and upper value as well. So in case of RMST, so RMST means root mean square deviation. So original value, original conformation is this one, the first one, and based on the, in comparison to the first one, the different conformation are generated, and you can just go through this. Okay, and you can check each and every conformations. Okay, and the RMST value, you can just look through it. So here you need to be clear about the RMST value as well. So RMST means root mean square deviation. So the value needs to be lesser than 2 angstrom for a perfect conformation. Okay, if it is away from the original pose, then it doesn't work in that sense. Okay, because it can have on specific binding, and a lot of other on specific sites would be incorporated into the interaction. So that is not feasible in nature. So there needs to be some kind of reliable interaction in the active site regions. So the RMST value of less than 2 is more favored. Okay, so you can see the first confirmation is okay, the second one is also fine, but the upper value is quite greater than 2. So okay, this one is perfect. Okay, the third one, two, three, four, fourth one is fine. Okay, and so on, you can screen it. Okay, the fourth one and first one looks better. Okay, let's check this first confirmation. Let's check the hydrogen bond of this molecule. So you need to select the intramodal. You can change the hydrogen bond coloration as well. Okay, based on what color you like, and you can label the hydrogen bond with distance as well. So here I need to make it clear about the hydrogen bond distance. Okay, so for biologically relevant, or you can say biologically significant interaction, the hydrogen bond distance needs to be lesser than you can say 3 angstrom, or better than better it would be lesser than 3 actually, but in many of the literature they have written 4 as well. So better you can see, better you can analyze the lesser than 3 angstrom hydrogen bond distance would be a better option. Okay, so you need to check for the hydrogen bond length as well if you are looking for biologically active complex molecule. Okay, so you just need to select the include intra-residue hydrogen bond as well, and now everything looks fine. You can just press onto OK. Okay, maybe running. Okay, fine. Okay, you can see in case of this conformation, okay, this conformation there are hydrogen bonds, but not in the first one. So you can go through the fourth conformation. It is found in the RMST as well. Okay, lesser than 2 angstrom. So there are 2 different hydrogen bond present over here. Okay, so you can check this, the length of hydrogen bond and everything. Okay, actually it's one only, it's 2.073 angstrom. Okay, so it looks perfect as well. If you want to check other confirmation, then you can just click here. Okay, there is no hydrogen bond in the first one. Second one, it has it doesn't have, okay, it doesn't have any hydrogen bond as well. The third one has hydrogen bond. Okay, let's see the fourth one. We have already labeled it. Fifth one, do pauses. Okay, so now let's screen the fourth confirmation. Okay, that's it. This we are focusing on. So now let's model this confirmation. Okay, so aspirin had already been selected, so let's label it. Okay, let's okay, it has not been selected yet, so you can press on to click. Okay, and then you can to press into upward arrow option. So it has been selected, aspirin has been already selected. Just you can go through select, and then you can create a zone of lesser than 5 angstrom. Okay, the zone is selected based on the favorable region. Okay, so for less than 5 angstrom region is more favorable for any kind of biological interaction, so that's why we select it. So okay, so it's a most favorable region in this case. Okay, now let's go to, okay, let's export some side chain molecule as well, side chain bases. Okay, so okay, these are the side chain molecules. Okay, it has a 2 hydrogen bonds, sorry. Okay, later on we will label it, don't worry. Okay, let's label the molecule as well. Okay, label it, residues. Okay, name and specifier. Okay, you can see it. Let's change the color of the label as well. Okay, color, all option. Okay, residue label. Okay, let's give it red coloration, yellow, green. Okay, make it red. Okay, fine. uh, Now you can see where it has been. You can see two hydrogen bond present over here. Maybe the valine. Okay, valine 250. Okay, 523 in A chain, and the hydrogen bond distance is 2.073 angstrom. It looks fine. And next, okay, let me check it. Okay, here it is. The hydrogen bond distance is 2.080, and it has been linked with serine 530. Okay, so there are two hydrogen bond for this particular conformation. Okay, in this way you can go through the labeling, and you can change the color as well. Okay, let's change the color of entire. Okay, you can see it. You can change the color of label only. Now let's go to color, and all option, residue label. Okay, what would be the best one? Magenta. Okay, magenta. And let's change the size of the label. Okay, fine. So you can see it has been well labeled, hydrogen bond distance. So you can also change the color of hydrogen bond distance and everything. So let's change the color of aspirin as well to make it look more. Okay, to provide a better contrast, let's change the color. Okay, go to color and then make it red. Okay, so you can see both the hydrogen bond over here 2.080 and 2.07, so it is perfect result. Now for the validation of your docking protocols, so in case of this particular molecule, it's a standard molecule, and it has parent ligand embedded into it, so you can easily to validate this, you can easily redock that parent ligand. For example, in case of this molecule, S5 is a parent ligand. You just need to go through you just need to go to PopChem and find the name of that S5 molecule. Okay, from PDB as well. From PDB, you can get the name, and then you can go to you can say PopChem to find the smile string and come back, and then prepare whatever we did before. Prepare that ligand molecule and then redock into the active site region. Okay, and then once you redock, you will get a confirmation of that parent ligand as well, best confirmation, where hydrogen bond will be there, because hydrogen bond is the most required bond for any kind of biological interaction. And it also depends upon what kind of ligand molecule you are using and what is the receptor as well, because in some cases hydrophobic interaction are also valid. Okay, so it depends upon what type of protein you are working on. So in this way you can redock that, and then after performing a after redocking that molecule, you will get one best confirmation. And with that best confirmation and the confirmation present in the database, you just need to superimpose both the molecules and then find the RMST value lesser than 2 angstrom. Once you find the RMST value less than 2 angstrom, you will get the perfect result. Okay, that means your docking protocol will be validated. And next step about validation of your docking protocol is to use okay, you can use positive control as well as negative control and do the molecular docking thing, and then you will be getting binding energy and everything there, interaction pattern. Just look for two things: binding energy and interaction pattern. In case of positive control, the binding energy will be the lesser binding energy will be present over there, and in case of minimum binding energy actually. And next, in case of positive control, in negative control, you will get a different result. Okay, and the interaction pattern would be different from positive to negative control. In case of positive control, it will be linked onto the key residues present in the pocket, while in case of negative control, it can have some kind of unspecific binding. So in this way you can validate your docking protocols. Okay, and there are other methods as well in case of validation, but I follow these two methods and I highly recommend them because it gives a reliable result. And I perform molecular dynamic simulation after doing this kind of you can say validation procedures, and so I got the best result in the molecular dynamic simulation. So you can also follow the similar pattern for validation thing. Okay, so this is it for the entire workshop. So if you have any questions, you can even save this molecule. Okay, save this entire complex and everything. So if you have any other question, then we will be moving on to Q&A session now. Thank you. So now let's deal with some of the questions that has been asked by Mr. Jamal. So he has asked that if no NA structure of protein is present in the database, then how can we analyze, how can you build the model and everything? So in that case, we can use homology modeling. You can develop your own model, okay, comparing to the standard protein present in the database, and then you just need to superimpose that standard structure or the standard structure of protein with your model. And then if everything is fine over there, if the RMS value is lesser than 2 angstrom, even lesser than 0.5, that would be the best one. Okay, and then you can also see the dimension, you can also see the dynamics of your chain and everything by using molecular dynamic simulation. Or else if you do not have that facility even, then you can just superimpose two of the protein molecule, okay, your model and a standard model. And if you get the desired RMST value lesser than 0.5 or even closer to 0, then that would be the best one. Okay, so in that way you can easily develop your model and then further analyze that result on. Okay, and next is he was also concerned about you can see uh, how the structure, okay, how we can predict the active site residues of non-standard protein or non-standard or non not fully solved structure in the database. So in that kind of protein molecule, you can get a sequence from the database. For example, in case in from the NCVI or even from any other databases. Okay, you just need to get the sequences and multiple sequences from different organism of the same protein, and then you can do multiple sequence alignment, and then you just need to see the conserved region. So the longest conserved region would be the active site, and in that case you can just mark the longest conserved region. For example, you can just mark the amino acid chain, amino acid residues, okay, from this to that, and then you can just mark it, and you can just come on to the visualization tool, and then you can just label those amino acid, and you can easily see the you can see active site residues and active site pocket as well. You can also create a pocket using visualization tools such as Chimera, or you can say Chimera, or any other tools, Discovery Studio Visualizer. Okay, so in this way you can easily screen that. Okay, and he has also asked that if ligand is bound to somewhere else, then what would be the option? So if you follow the exact step, okay, if you follow the proper step, then you would not find any unspecific binding. Or in case of this kind of result, okay, if you find any unspecific binding nature of your ligand molecule in the pocket, then that is not a valid result. So you just need to trace back your protein as well as your model, and then you just need to superimpose again, and then look for the you can say Ramachandran plot as well. Look for the favored region if it is quite better, okay, if all of the residues fall under the favored region or not. Maybe more than you need to look for more than 90% of favored region, and you also need to look for the mole probability score and all that thing. Okay, once it is validated, then only your result will be valid result. Okay, everything is sorted in that way. So I think that's the end of workshop. Actually, we made this, you can say we re-recorded this video because yesterday we had some kind of sound issues on YouTube, so we couldn't face that stream into the YouTube with proper sound. So we made we re-recorded this video, and we are going to upload that on YouTube as well. So you can compare both the videos, okay, both the videos, and you can work on any kind of you can say mini projects or even on a projects. And if you find any kind of queries, or if you have any kind of queries, then you can just write us, you can send an email, we can work together. You can also join us in any kind of further workshop or webinars. We are planning to organize one intensive program, a virtual intensive program regarding computer drug designing, where we'll be covering from basic bioinformatics to molecular docking, and beyond molecular docking even. So you can join that course as well. You just need to pay some minimal charges for that, and then you can get a full benefit from the courses, okay, because it's just a 1 or 2 hour courses. Then we can give you best out of it, and then you can join this kind of program, okay, and Silico programs that is on the line. And then in the future as well, we will be organizing different webinars, different workshops regarding computer drug designing, as well as different bioinformatics program are on the line. So you can join us. You can get you can get benefit from this kind of program as well. So hope to see you around in those kind of programs. Okay, this is all for today. Okay, thank you everyone.